Very dear students, today we are going to discuss about cell cycle. The cell cycle or cell division cycle is a series of events that take place in a cell leading to its division and duplication. The cell cycle may also be defined as an ordered cycle of complex events resulting in cells proceeding to cell division from a resting state. The cell division cycle is a carefully choreographed series of events that culminates in cell division. The fundamental task of the cell cycle is to faithfully replicate DNA and to equally distribute identical chromosome copies to two daughter cells. Genetic defects affecting the cell cycle machinery contribute to uncontrolled cell division, the hallmark of cancer. The length of the cell is important because it determines how quickly an organism can multiply. For single-celled organisms, this rate determines how quickly the organism can reproduce new independent organisms. For higher order species, the length of the cell cycle determines how long it takes to replace damaged cells. The duration of the cell cycle varies from organism to organism and from cell to cell. Certain fly embryos sport cell cycles that last only 8 minutes per cycle. Some mammals take much longer than that, up to a year in certain liver cells. For fast dividing mammalian cells, the length of the cell cycle is approximately 24 hours. So there are two major phases of cell cycle: interphase and mitosis. So first of all, interphase. Interphase is the holding stage or stage between two successive cell divisions. It is the phase of the cell cycle in which the cell spends the majority of its time and performs the majority of its purposes, including preparation for cell division. In preparation for cell division, it increases its size and number of organelles and makes a copy of its DNA. Interphase is also considered to be the living phase of the cell, in which the cell obtains nutrients, grows, reads its DNA, and conducts other normal cell functions. The majority of eukaryotic cells spend most of their time in interphase. Interphase does not describe a cell that is merely a resting, but is rather an active preparation for cell division. The duration of time spent in interphase and in each stage of interphase is variable and depends on both the type of cell and species of organism it belongs to. Most cells of adult mammals spend about 20 hours in interphase. This accounts for about 90% of the total time involved in the cell division. Now there are four stages of interphase. Each phase ends when a cellular checkpoint checks the accuracy of stage's completion before proceeding to the next. The stages of interphase are G0 stage or rest phase, G1 stage or first gap phase, where the cells prepare to synthesize DNA and undergo mass synthesis, S stage, where the cells synthesize DNA and double chromosome number, G2 stage or second gap phase, where the cells prepare to divide. Now first of all, G0 or gap phase of cell cycle. During G0 phase, cells withdraw from the cell cycle and are dormant and do not grow or divide. This is a way for multicellular organisms to control cell proliferation. The time cells spend in G0 and the specific signals needed to move the dormant cell back into the cell cycle vary greatly depending on the type of cell. Cells can remain in this phase for days, weeks, or even years. Most of the cells in multicellular organisms, including humans, are currently in this phase. For example, muscle and nerve cells are permanently in a state of G0 phase. Liver cells also remain in this phase unless they are stimulated to grow after an injury. The term post-mitotic is sometimes used to refer to both quiescent and senescent cells. No proliferative cells in multicellular eukaryotes generally enter the quiescent G0 state from G1 and may remain quiescent for long periods of time, possibly indefinitely. This is very common for cells that are fully differentiated. Cellular senescence is a state that occurs in response to DNA damage or degradation that would make a cell's progeny non-viable. It is often a biochemical alternative to the self-destruction of such a damaged cell by apoptosis. At a certain point in G1 phase, the cell monitors internal and external environments to determine if it should go through the entire cell cycle and divide. If the cell senses that there are not enough nutrients such as amino acids or growth factors for division, then it often enters into G0 phase. During this time, normal cellular activities are drastically reduced. For example, protein synthesis is inhibited by 50 to 80%, and many proteins are degraded. Enzymatic activity and RNA synthesis are also severely inhibited. Cells in G0 phase can quickly re-enter G1 phase and progresses through the cell cycle if they receive signals that nutrients are available for cell division. Now, G1 or gap first phase of the cell cycle. The G1 phase is a period in the cell cycle during interphase, after cytokinesis and before S phase. For many cells, this phase is the major period of cell growth during its lifespan. During this stage, new organelles are being synthesized. So the cell requires both structural proteins and enzymes, resulting in great amount of protein synthesis and high metabolic rate in the cell. Now, dear students, let us see the stats of the genome. The DNA in a G1 diploid eukaryotic cell is 2N, diploid state, meaning there are two sets of chromosomes present in the cell. The genetic material exists as a chromatin, and if it were coiled into chromosomes, there would be no sister chromatids. Haploid organisms, such as some yeast cell, will be in haploid state and thus have only one copy of each chromosome present. There is a restriction point present at the end of G1 phase. This point is a series of safeguards to ensure that DNA is intact and that the cell is functioning normally. Functionally, the safeguards exist as proteins known as cyclin-dependent kinases. In abbreviated form, we call them CDK. S-phase promoting factor. In abbreviated form, we call them SPF. Now, the G1 CDK proteins activate the transcription factors for a variety of genes. These include genes which are responsible for DNA synthesis, proteins, and S-phase CDK proteins. Now, S-phase of cell cycle. The S-phase is a period in the cell cycle during interphase between G phase and the G2 phase. Following G1 phase, the cell enters the S stage when the DNA synthesis or replication occurs. At the beginning of the S stage, each chromosome is composed of one coiled DNA double helix molecule, which is called a chromatid. The enzyme DNA helicase splits the DNA double helix down the hydrogen bonds. DNA polymerase follows, attaching a complementary base pair to the DNA strand. making two new semi-conservative strands. At the end of this stage, each chromosome has two identical double helix molecules and therefore is composed of two sister chromatids joined at the centromere. During S phase, the centrosome is also duplicated. These two events are unconnected but require many of the same factors to progress. The end result is the existence of duplicated genetic material in the cell, which will eventually be divided into two. Damage to the DNA often takes place during this phase, and DNA repair is initiated following the completion of replication. Incompletion of DNA repair may flag cell cycle checkpoints, which halts the cell cycle. However, after the cell has completed this phase, it is very likely that the cell will continue on to complete and the cell cycle, and the cell that is not due to divide will not go through an S phase. Rates of RNA transcription and protein synthesis are very low during this phase. An exception to this is histone production, most of which occurs during S phase. Now, gap second or G2 phase of cell cycle. The cell then enters the G2 phase, which lasts until the cell enters mitosis. Again, significant protein synthesis occurs during this phase, mainly involving the production of microtubules, which are required during the process of mitosis. Inhibition of protein synthesis during G2 phase prevents the cell from undergoing mitosis. It is a relatively quiescent part of the cell cycle during interphase, lasting from the end of DNA synthesis, the S phase, until the start of cell division, the M phase. Now, M phase of the cell cycle. Mitosis is the process in which an eukaryotic cell separates the chromosomes in its cell nucleus into two identical sets in two daughter nuclei. It may also be defined as the type of cell division within the body where the cells divide into other cells, each with a full set of chromosomes. Each of these cells receive an exact copy of chromosomes in the original state. During development, mitosis occurs again and again until finally the adult organism is created. When an eukaryotic cell divides into two, each daughter or progeny cell must receive a complete set of genes. For diploid cells, this means two complete genomes or 2N, a pair of centrioles in animal cells, some mitochondria, and in plant cells chloroplast as well, some ribosomes, a portion of endoplasmic reticulum, and other organelles. The process of mitosis is a complex and highly regulated. The sequence of events is divided into phases corresponding to the completion of one set of activities and the start of next. The M phase has been broken down into several distinct phases, subsequently known as prophase, prometaphase, metaphase, anaphase, telophase, and cytokinases. Well, dear students, let's first of all discuss about prophase. The prophase is the first stage of mitosis, during which chromosomes condense, the nuclear envelope disappears, and the centrioles divide and migrate to opposite ends of the cell. Now, let's briefly see what changes take place during prophase. The two centrosomes of the cell, each with its pair of centrioles, move to opposite poles of the cell. The mitotic spindle forms. This is an array of spindle fibers, each containing 20 microtubules. Microtubules are synthesized from tubulin monomers in the cytoplasm and grow out from each centrosome. The centrosomes become shorter and more compact. The nuclear membrane starts disintegrating. Now, pro-metaphase. Pro-metaphase is the phase of mitosis following prophase and preceding metaphase in eukaryotic somatic cells. Some changes that occur in a cell during pro-metaphase are: first, the nuclear envelope disintegrates because of the dissolution of lamins that stabilize its inner membrane. Then, a protein structure, the kinetochore, appears at the centromere of each chromatid. With the breakdown of nuclear envelope, spindle fibers attach to the kinetochores as well as to the arms of chromosomes. Failure of kinetochore to become attached to a spindle fiber interrupts the process. Now, next phase we have, which is metaphase. Metaphase term comes from the Greek meta, meaning after. As microtubules find and attach to the kinetochores in the prometaphase, the centromeres of the centrosomes convene along the metaphase plate or equatorial plate, an imaginary line that is equidistant from the two centrosome poles. This even alignment is due to counterbalance of the pulling powers generated by opposing kinetochores, analogous to a tug of war between people of equal strength. Now, some changes take place during metaphase, and they are: number first, the nuclear membrane disappears completely. Number second, in animal cells, the two pair of centrioles align at opposite poles of the cell. Number next, polar fibers, the microtubules that make up the spindle fibers, they continue to extend from poles to the center of the cell. Chromosomes move randomly until they attach at their kinetochores to polar fibers from both sides of the centromeres. Chromosomes align at the metaphase plate at right angles to the spindle poles. Chromosomes are held at the metaphase plate by the equal forces of polar fibers pushing on the centromeres of the chromosomes. Now next we have anaphase. Anaphase, which has been from the ancient Greek ana, meaning up, is the stage of mitosis when chromosomes separate in an eukaryotic cell. Each chromatid moves to opposite poles of the cell, opposite ends of the mitotic spindle, near the microtubule organizing centers. Anaphase begins abruptly with the regulated triggering of the metaphase to anaphase transition and accounts for approximately 1% of the cell cycle's duration. At this point, the anaphase becomes activated. Now, some changes that occur in a cell during anaphase, they are: number one, the paired centromeres in each distinct chromosome begin to move apart. Next, one is the paired sister chromatids separate from one another. Each is considered a full chromosome. They are referred to as daughter chromosomes. Next, through the spindle apparatus, the daughter chromosomes move to the poles at opposite ends of the cell. Next, the daughter chromosomes migrate centromere first and the kinetochore becomes shorter. In preparation for telophase, the two cell poles also move further apart during course of anaphase. At the end of anaphase, each pole contains a complete compilation of chromosomes. Now we have one more phase, which is telophase. Telophase name derived from the Latin word telos, which means end, is a reversal of prophase and prometaphase events. It may be defined as period wherein the chromosomes arrive at the poles, the microtubules disappear, and the nuclear envelope reappears. At telophase, the non-kinetochore microtubules continue to lengthen, elongating the cell even more. Corresponding sister chromosomes attach at opposite ends of the cell. A new nuclear envelope using fragments of the parent's cell nuclear membrane forms around each set of separated sister chromosomes. Both sets of chromosomes, now surrounded by new nuclei, unfold back into chromatin. Some changes that occur in cell during telophase, they are: number first, the polar fibers continue to lengthen. Number second, nuclei begin to form at opposite poles. Number next, the nuclear envelopes of these nuclei are formed from remnant pieces of parent's cell's nuclear envelope and from pieces of the endomembrane system. Number next, nucleoli also reappear. Chromatin fibers of chromosomes uncoil. After these changes, telophase is largely completed and the genetic contents of the cell have been divided equally into two. Now we have cytokinases. Cytokinases, the name derived from Greek cyto, means cell, and kinases, means motion or movement, is a process in which the cytoplasm of a single eukaryotic cell is divided to form two daughter cells. It may also be defined as an organic process consisting of division of the cytoplasm of a cell following karyokinases, bringing about separation into two daughter cells. Cytokinases must be temporarily controlled to ensure that it occurs only after sister anaphase separation during normal proliferative cell divisions. To achieve this, many components of the cytokinases machinery are highly regulated to ensure that they are able to perform a particular function at only a particular stage of the cell cycle. So, dear students, this is all about cell cycle. Here we have discussed about mitosis, various stages of mitosis, various changes that take place during different phases of mitosis. Thank you very much.